Wonderful. I'd like to welcome everyone to the SCAR Open Science Conference Session 18, entitled Management Implications of Southern Ocean Ecosystem Dynamics and Biodiversity Thresholds. And thank you so much for participating. This session is critically important in relation to connecting the ecological processes we're observing in the Antarctic marine ecosystem to direct responses that humanity has some control over, with the goal of maintaining the relationships between components of the Antarctic ecosystem. The relationship between management and ecosystem dynamics and thresholds can be coordinated and complementary, but most optimally through a system of feedback management. So this session is divided into two sessions, this being the second one, and it will last 2 hours, during which we will have 8 talks followed by a discussion. And these talks, really excited about them. They cover a wide variety of topics, including spawning grounds for Antarctic fish, the Ross Sea ecosystem, parasites, and many talks focused on Antarctic krill and penguins, including those on potential impacts of climate change and fishing. With that, I'll pass it over to Jilda to start us off.
Thank you, Cassandra. And I just wanted to mention, actually, it's really a privilege to be convening this with Cassandra this afternoon, because we first met in person in 2016 at the SCAR OSC in Malaysia, and we co-presented a talk, and now we're co-convening a session. So this is really cool for it to my perspective. So just a couple of housekeeping notes. So speakers, when you're not speaking, feel free to turn your video off. And the talks are supposed to be 8 minutes with a wiggle room of 10 minutes, but we won't be too strict on that because we do have a bit of time, but we don't want to eat too much of our discussion period, which is just 30 minutes with the 8 talks. So if you go a bit over 8 minutes, we might save questions for your talk for the discussion section. And attendees, remember that you can put your questions into the Q&A box. Okay, perfect. I will do that for Virginia. So you can put your questions into the Q&A box, and then we can pose them to the speakers to answer live, or the speakers can type in their answers, during the next talk if we need to get things moving. So just a reminder about that. Okay, perfect. Virginia just was added to our last speaker. Sorry about that. Yeah, Virginia, real quick, do you want to double check that you can share your screen before we get started? Okay, we see
Can you see the
PowerPoint.
screen?
We see PowerPoint. Do you want to try starting the presentation?
Yes.
I love how we all have the same toothfish pictures. I keep seeing the same pictures in all the talks. Perfect. We see the presentation.
Thank you.
Thank you so much, Virginia.
Thank you.
Okay, with that, I'd like to get started with our first talk and introduce Manuel Novillo, who's going to talk to us about inshore spawning grounds of notothenioids. Manuel, you have the floor.
Thank you very much. So while I'm sharing, I wanted to thank everyone, the organizers, for all the organization, and of course, hoping to meet you in person next year. So I don't know if it's everything okay, Jilda? It's going on?
Yes, we see the presentation, and now if you start, perfect, we see the presentation view.
Great. So, well, as you said, my name is Manuel Novillo. I'm from Argentina, and I will be speaking a bit about the role of inshore areas as spawning grounds in Antarctic notothenioids. Just briefly, these are some general reproductive features of this fish group. They have a delay on the sexual maturation. They spawn once a year. There's also a prolonged gametogenesis, and they produce a few eggs of large size. So that is to say, it takes them too long to lay few eggs, and they do it once a year. So we think these reproductive features are drivers that explain an amazing behavior, that's the parental care behavior, which has been summarized by Mario Lamesa and colleagues recently. And apart from being so interesting, this behavior, it gives us some extra information regarding the spawning substrate. That is to say, the substrate in which the fishes are choosing to lay their eggs. In line with this, this particular issue, the spawning ground location, is one of the major knowledge gaps at this moment for these species. Most of the research produced try to define or locate these spawning grounds, but some phrases are quite vague. So they spoke about inshore waters, offshore waters, or rather on the slope, and have been mainly focused on commercial exploited species. That data arising from offshore surveys where spawning individuals have been identified or egg and larvae occurred on their catches provided information to try to define a spawning ground location. Recently, the use of ROVs have provided further insights into this research area, while this picture has been widely distributed and is one of the most amazing discovery in the last years of this vast breeding colony of these icefish at the Weddell Sea. But nevertheless, the information that comes from inshore localities is still limited. So I want to present the ichthyology project of the Argentinian Antarctic Institute that has been working and performing research for more than 30 years in what it is a long-term shore-based sampling, mainly at Potter Cove. And this provided valuable information to CCAMLR in order to assess the impact of former commercial fisheries, offshore commercial fisheries, the impact they had on inshore fish population of notothenioids. This project has also expanded recently to new areas as Paradise Bay at West Antarctic Peninsula. So I will summarize what we have obtained in terms of the reproductive biology of this fish group. So this is Potter Cove at the South Shetland Island, where Carlini Station is located. So let's zoom in a bit on the cove. This is a picture, a graph that shows a ridge that divides the cove in an outer zone, which bottom is covered by rocks, by macroalgae. There are also sponges, lots of invertebrates. Here, the depth gets up to down to 120, 150 meter depth. So there's a picture for you to see what's going on down there. And on the other side, we have the inner zone where the sediment is a muddy sediments full of drop stones and algae are scarce. And this area reaches down to 60 meters depth. This inner zone is mostly product of a retreating glacier, which is called the Fourcade Glacier. At this site, there was in situ observation of the parental care behavior of Paracagnitis charcoti, which is a dragonfish. And also there we had obtained several evidences from microscopic, histologic, and physiologic approach that it's a spawning site, of course, for this dragonfish, and strongly suggests that it's a spawning site for Cainocephalus aceratus and Nototheniops nidifrons, other notothenioids. As well, Trematomus bernacki, we suggest that it's spawning on the outer zone where the sponges are located, and it is known that this species uses the sponges to lay their eggs. Moreover, there are evidences that also Notothenia cordyceps and Trematomus eunensis should be spawning at this site. Of course, we also found Arbachifera antarcticus. This species lives the entire life cycle in shallower waters and also uses the cove for spawning. The case of Potter Cove is not an isolated case, and I wanted to highlight this. We also found similar evidences at the West Antarctic Peninsula, where Paradise Bay is located, in particular in a cove called Alvaro Cove that reaches down to 100 meter depth. So these three species I'm showing are spawning at the site. And recently we also found a stomach of Notothenia cordyceps full of a mass of eggs that we could identify through molecular techniques as being of Trematomus bernacki, which informs us that and confirms that this species also spawn at the cove. In addition to this, we had several catches of Trematomus hansoni, gravid females. As you can see, these are fully hydrated ovaries that indicates us that the spawning was imminent at the same site. So I wanted to show this graph, which it spots the different factors that might be shaping and framing the reproductive cycle in fishes. You can see that photoperiod and temperature have greater importance at higher latitudes, as it occurs in Antarctic waters. And these two reproductive factors act as environmental reproductive cues, because they produce changes in the endocrine pattern that precedes the ovarian processes we usually observe and we usually examine from a macroscopic and histological examination. So I wanted to tell you this because when trying to understand what's going on at shallower waters, we believe that the impact that these two environmental factors have is greater. So it's easy and it's logical to think that the impact that sunlight has on the shallower water is greater than the one it has at deeper waters. So in terms of the change of photoperiod phase, this environmental factor might be leading to changes on the endocrinology of the Antarctic fishes, as well as the temperature. We know that the thermal inertia at shallow waters is greater, so changes in water temperature might be drastic. Our data also suggests that these two species, Baragagnictis charcoti and Cainocephalus aceratus, might be experiencing an inshore spawning migration to inshore sites. Of course, this is interesting because they might be a selection of sites where to spawn. So I wanted to highlight in this short lecture that our data reinforce the role that inshore localities have as spawning grounds for Antarctic notothenioids, as they might be providing several different kinds of substrates for them to lay their eggs. They also play a role of nursery grounds as they protect early larval stages and early juvenile stages and provide food as well. What I wanted to highlight also that in this global warming scenario, we need to deepen the role that the environment has in the reproductive spatiotemporal window delimitation to know what is going on and predict future changes as we might expect on this actual global warming scenario. All this data that is produced by our ecology project had provided in the past and continues providing valuable information in scope to CCAMLR, in scope of the conservation of this fish population and the marine ecosystems, as the protection of fish reproductive ground is one of the most successful management strategies nowadays. So I wanted to thank all my colleagues and my group. Of course, we are for the scientific issues we always develop, but also because we are a great human group. And I leave these questions to encourage the discussion if we have time. Also, there's my the way to contact me, my email, and all these institutions that have been involved. I want to thank you very much.
Thank you so much, Manuel. We have a question for you, but we're going to save it to the discussion time in the interest of timing. And we also want to say that we loved your animations and your photos. So thank you again for that. And I think we'll move right along. And I'd like to introduce Virginia, who is going to be talking to us about the resurgence of the Anthropocene in the Ross Sea. So please, Virginia, you have the floor.
Thank you. Okay, first, thanks everyone for coming to my talk. And I'm Virginia Morandini, currently working at the Spanish National Science Museum. And yes, I'm going to talk about the resurgence of the Anthropocene in the Ross Sea in Antarctica. And the Ross Sea, it has been nicknamed the last ocean. And that's because in 2008, some authors mapped over 232 sectors of the world oceans using 17 anthropogenic factors that affect marine ecosystems. And they found that the Ross Sea was the sea that exhibited the last total impact. So it was like a pristine area according to the period that they were analyzing. So here I would like to review the historical impacts to what we call the indicator species that are present at the Ross Sea, and which are the populations and trends, including the emperor and Adelie penguins and the weddell seals. After that, I would like to review also the decadal time series of the ocean and climate variables, and as well as fishing pressure of a trophic competitor, the Antarctic toothfish. And I will try to explain you which hypotheses we had to reveal what has been changing. And after, I will show you which modeling we did in trying to identify which factors explain these species trajectories. So before to start, just to show you a really simple trophic chain that's happening at the Ross Sea. So just to highlight that here at the bottom, we have an Antarctic silverfish that is the common prey of many different species. For example, the emperor Adelie penguins, the weddell seal, but also the toothfish. So for example, Adelie penguins, they don't only prey on the Antarctic silverfish, but also they prey on the krill, and the weddell seals, for example, they have two different preys, also the Antarctic silverfish, but also the toothfish. And the biggest toothfish, they can prey also on Antarctic silverfish. So there is a food chain that has at the bottom the Antarctic silverfish. So we have focused our work on the changes of three different species, the emperor penguins, the Adelie penguins, and the weddell seals. And those species, according to the Commission of the Conservation of Antarctic Marine Living Resources, have been called indicator species because are sensitive species to changes in the ecosystem and are at the top of the food chain. So for the three species, the population has been monitored for several decades, and they have been stable for a couple of decades, but more recently have been dramatically increasing. And that seems to show that the ecosystem structure on the Ross Sea has been changing. So, for example, here we can see the emperor penguins. We didn't count, the data that we have is not from the direct count of the emperor penguins, but from the emperor penguin chick, assuming that every chick has two adults. And this is the trend that we can see that have been increasing. Of course, we have to not to take in account those years that are here highlighted in blue from 2000 more or less to 2005, because during that period, two huge iceberg grounded near the Ross Island, changing all the sea ice movement, and some of the adults, they couldn't get to the water to forage, so the productivity was extremely low. And so we talk out those years in our analysis because the presence of this mega iceberg, but the emperor penguins at the Ross Sea is the 28% of the world population, and that's been increasing since 1998. The same for Adelie penguins. Here we consider Adelie penguin pairs. The counts has been done in December, and also you can see that the Adelie penguin pair has been increasing since 1998. And we consider three different colonies: the colony at Cape Crozier, Cape Bird, and Cape Royds. However, for those analyses, we only consider the colonies at Cape Crozier and Cape Bird that has been increasing, because it seems that the colony at Cape Royds didn't recover after the mega iceberg event. And analyzing that colony, there are so many variables that are affecting the recovery, including the small size. We decided to do not include this colony in this analysis here. And finally, we analyzing the trend of what the weddell seals. The weddell seal population at the Ross Sea is the 42% of the world population. And also, we counted not directly the adults, but the pups during December too. And the annual pup count has been dramatically increasing, as you can see here in the graph. Also, the weddell seas has been affected by the iceberg, so we didn't include those years in the analysis either. So what happening with biophysical variables related with biophysical environment? So we analyzed the sea ice extent. The trend has been to increase over the years. Also, and how this can affect, for example, Adelie penguins population. So it may affect the recruitment. So higher sea ice extent, it can take for the penguins longer to come back to the colonies for breeding. For the Ross gyre speed, for example, it has been increasing too over decades. So here you can see the Ross Sea and his position related with the Ross ice shelf. And when this gyre increasing speed is affect different colonies of penguins. For example, it affects the wintering period. So it affects the probability of the penguins to be able to come back to their own colonies after wintering to breed. For those winters in the eastern portion of the gyre, the spring arrival is easier where the gyre speed is higher because it pushed them to their own colonies. But for those penguins wintering in the western portion of the gyre, the higher gyre speed would have a negative effect as the penguins must travel contrary to the ice movement. And other variables that we analyzed was, for example, the mean open water that has been increasing to the mean sea surface temperatures that has been increasing to. This surface temperature might affect the stratification of the column of the water. So when increased, it may be more easier for penguins to catch their own prey because the water is not it's more clear, we can say. But the primary production and the chlorophyll didn't have significant changes over the years. So the production of the Ross Sea looks like didn't change over the period that we analyzed it. And other interesting variables are the main air temperature. So that has been also increasing, and that can affect the proportion of the colony that the penguins can use for breeding, and the mean wind speed, that it can be related with so many different variables, that the sea ice extent or the or even the sea ice temperature. In addition to those physical variables, environmental variables, we wanted to add other variables related with fishing. So since 1999, the fishing to the toothfish started at the Ross Sea and has been slightly increasing since it started. So here we can see on the left the percentage of fish by the total length. So we divided smallest toothfish that has been caught by commercial fisheries and the largest one. So we can see that the largest one has been decreasing because the prevalence in the water. So fisheries, they will also try to catch the largest toothfish as possible, but looks like over the last years, the presence of those toothfish has been decreased because they has been depleted probably. And with all these variables and those data, we included the annual growth of all the colonies studied and in an ARIMA model. We built different models following model set construction rules, and we selected the best model for each set according AIC criteria. So for Adelie penguins at Cape Crozier, for example, the best model included these 4 different variables. So a negative correlation of the annual growth of the Adelie penguins at Cape Crozier with the gyre speed and the open water date. So that's a later date when the open water gets the 50% of the total open water, and the gyre speed are negatively correlated with the annual growth. However, the cumulative fish is 3 years cumulative fish of big toothfish, and the air temperature are positive correlated. So with a higher number of high and big toothfish catch during commercial fisheries, the annual growth increases. And with higher air temperature, the annual growth increases too. This is the best model set for Cape Bird, the Adelie penguin studied at Cape Bird, and we can see a negative correlation also with open water date, and we see ice extent, and a positive correlation, however, differently to what we found at Cape Crozier with the gyre speed, and again, as we found at Cape Crozier, a positive correlation with the cumulative fish, so the extraction of the largest toothfish by the commercial fishery. And finally, for the Weddell seals, the best model included a positive correlation with the fish size extent, again, with the cumulative fish extracted by the commercial fisheries, with the open water extent, and a negative correlation with the gyre speed. So looks like even with decades of ocean weather variables changing, like the variables that we analyzed here, penguins and seals have been increasing only after fishery initiation in late 1990s. So what we found is that a common variable that included the best model of both penguins and seals is the cumulative fish. So the cumulative catch of toothfish larger than 134 centimeters. So the toothfish the large toothfish have a really slow demography and is not easily replaced in the water. So this increase in the number of penguins and seals that in most cases can indicate a successful management of the population here might indicate that the ecosystem is changing. Those big toothfish that have been fished may not be replaced, and this is changing the structure of the ecosystem that has been present so far. For example, the reduction of those toothfish is affecting the presence of silverfish in the water. That is the main prey of penguins and seals. Because of that, the populations that we are studying of penguins and fish and seals are increasing, because as fishery is impacting toothfish is decreasing, silverfish is increasing, and the populations that prey on silverfish are increasing, as we said before. So what we suggest here is that the column food web is changing. The silverfish is increasing because this extraction of the big toothfish, but that is not probably reflecting that we are something positive to the indicator species, but a change in the ecosystem. So we suggest that management should be reevaluated in keeping with the rational use ecosystem-based viewpoint. So we should probably try to understand better what's happening in the ecosystem, why these indicators per species are increasing, and what that means for the whole ecosystem. So I want to thank you, all the co-authors that are helped in the analysis, writing, and provided the data. And thank you all for attending to this talk.
Thank you so much, Virginia. In the interest of time, we're going to move right along, but we've got some interesting questions for you for the discussion portion. So I'd like to pass the baton over to Thomas Devigne, who will be talking to us about parasite outbreaks. So thank you, Thomas, you have the floor.
Thank you. Okay, so Do you see my PowerPoint there?
Yes, all good.
Cool. So yeah, I'm Thomas Devigne. I'm at the University of Oregon in the USA. And today I'm going to present you some multidisciplinary collaborative work that we recently published about a parasite outbreak that we observed in notothenioid fish on the Antarctic Peninsula. So in a changing environment with parameters such as temperature, salinity, or pH that are changing, these can create a direct abiotic stress on fish. And a lot of people, including us, study that. But those same changes in the environment can have impact on other components of the ecosystem, like viruses, parasites, and that can influence species communities, trophic interactions, which in return can create a biotic stress on the fish. And this is what I'm going to present you today. One night when we were fishing in Envord Bay, and we collected a lot of specimens from that given species, Traumatomys scotti, but a lot of them had this type of like massive skin tumors. So we really wondered what that was, and so that's what I'm going to present you today. But if you want to know about this study in a different angle, we created a short scientific graphic novel with undergraduate student Chloe Damonio. And you can just scan this QR code and it's going to take you to the page. We have it in 5 languages, and we will take you about the Gould at Palmer Station and back in the US for the analysis. And I'm also going to present that on session 49 on Wednesday, if some people are interested. But today I'm going to present you the scientific part of it. So we were fishing in Envord Bay, not too far from Palmer Station, and we captured hundreds of specimens of that species, and about 30% of them had those big tumors that could be located anywhere on the body. A week later, at another fishing site in Dolman Bay, we captured one specimen of another species that had also big tumors. When we came back to the States with some samples, we asked a lot of Antarctic fish researchers we know if they've ever seen that, and 10 told us they've never seen that before. 4 told us that they've occasionally seen something similar. So from that survey, we concluded that what we observed was a recent outbreak of a disease that may have been present, but rare actually in the past. So what can be causing these tumors? For that, the first thing we did was to do a metagenomic analysis of tumor and healthy skin. So we did a lot of sequencing for 6 samples of each group, and we assembled them and taxonomically classified them based on which group of life they may most likely come from. And what we observed in those 6 tumor samples that are the 6 bars on the right is that one group, the microbial eukaryote, is really abundant in those samples compared to the healthy skin. Meanwhile, we were also working with pathologists, and we observed that there was abundance of unicellular alveolates that are referred to egg cells. And so they are those pink, light pink color patches within the dermis, or here the light pink, whereas the darker purple is host cells. So with this diagnostic, I was able to design some PCR primers to amplify the 18S gene of XL and confirm that indeed there was a lot of XL DNA in the tumor samples while it was absent from the healthy skin samples. So our conclusion was that the fish we encountered were suffering from XL disease. So you probably wonder, what is XL disease? So it's caused by a parasitic unicellular alveolate of the Exelidae family. It manifests into parasitic xenomas in various fish groups throughout the world, from Norway and Iceland to actually Antarctica, where there are several cases reported. And they can touch the pseudobranch, the epidermis or the gills themselves. And actually, all the cases reported in Antarctica so far have been in the gills, like this case in the Ross Sea, and not in the skin, like we observed. So we wanted to know what was the XL that was causing those tumors in those fish. So I gathered all the 18S rDNA sequences I could find, and all our sequences indeed like grouped within the Exelidae family. And within that group, our sequences that are here in Notoxelia were forming a very well individualized subtree, which allowed us to describe a new genus of XL parasite that we call Notoxelia in reference to the type host notothenioid. And within this group, actually the sequence from the second species was very different, or different enough for us to be able to differentiate two species of Notoxelia. So now that we have an identity for the parasite, how do XL grow and spread on the fish? So we used some molecular histology method first, in situ hybridization, where we did sections, and by methods, we stained in blue all the XL cells and counterstain in red what is fish cells. And what we see is that XLs actually develop and proliferate and fill large compartments between very thin layers of fish cells. So in the end, the skin is almost entirely XL. When we look at how much it can spread on the fish, for that we use a randomized grid point where each point or intersection of the grid on the fish were classified as either healthy skin or affected skin. And by combining both sides of fish for more than 20 fish, that allowed us to create a prevalence map where we actually see on this rough outline of a fish, the average, that we can find those tumors or affected skin anywhere on the body, actually, but they are more frequent behind the head and close to the anus. And that allows us to hypothesize that the infection might be linked to feeding, or actually to simple contact with the substrate. So how does that impact the fish itself? So we use micro-CT and micro-MRI, which allowed us to create some 3D models and actually go very in fine details and see that the parasites did not invade the body. They really grew within the skin and stayed in the skin. But what we observed in this specimen at the tail is that within the underlying muscle, new blood vessels have formed. And we could model those as well, separate them in 3 dimensions, and show that those blood vessels were actually irrigating the tumor itself. And those blood vessels are really not normal on a normal fish tail. So they really, the tumors really take from the fish itself. So what we did next was to compare the length-weight relationship between fish that are healthy or visually healthy with fish with obvious tumors. And what we saw is that the growth curve of fish with tumors was lower than healthy fish. And in the end, when we normalize by the length, it turns that fish with tumors were on average about 10% lighter than their expected weight if they were healthy. So in conclusion, what we found in this case was that we have a novel XL parasite that caused an unprecedented disease outbreak in Antarctic fish and detrimentally affected the fish health. So there's a lot of questions that can arise from that. Like we can wonder whether the fish was getting weaker because of abiotic stress on it, and then like getting sick more easily. Or actually the contrary or complementary. Could it be that those new conditions in the environment are making XLs more virulent, more proliferative, and create this outbreak? But actually, it could also be something completely different. It doesn't have to be related to climate change. Maybe this parasite has a very long life cycle, and let's say manifests into an outbreak every 20 years, and we just happen to be there at that moment when the outbreak was happening. There's really a lot of questions that remain. And for that, we need a better survey year after year, if possible, to be able to like monitor the production of these diseases and maybe other diseases. And one of the big questions I have is that whether this outbreak is continuing to spread to other places and maybe start to infect other species. And so I'm going to finish today with the last page of our short graphic novels and to say that, yes, we are planning to go back and to explore additional areas to try to answer these questions. So on this, I'm going to thank all the co-authors on this project from many different places and many different skills. Thank you.
Thank you so much, Thomas. Super cool about the graphic novel. That's amazing. It'll be interesting. I hope folks will be able to get to see your talk more specifically on that next week. So just to switch gears a bit, so we had two very interesting and different fish talks to start our session off. And now we're going to be switching gears to more krill-oriented work. So I'm going to pass the baton over to Cassandra to start introducing that sector of the session.
Yeah, thanks so much, Jilda. And again, thanks to everyone for the great talks. Thomas, same thing. I actually would love to hear more about the graphic novel and what a great way to spread the word about amazing Antarctic fish. Yeah, so as Jilda mentioned, we're going to pivot to hearing a lot about krill and some of their predators in these next 5 talks. So I'd like to invite Mauro Mardones to go ahead and kick us off with the first krill talk.
Yeah, I'm going to share my screen. You can see?
Yes, it looks great.
Yeah, thanks for this opportunity. My name is Mauricio Mardones. The name of this talk is applying integrated stock assessment model to diagnose exploitation status and population variable in Antarctic krill. The research motivation start because today the krill management featuring is based on acoustic surveys and no reference point or climate change driver are used to biomass estimation and the management. This lack and this information has effect on the long-term scenarios in the krill stock. So the main objective is to use a stock assessment model that propose reference point based on MSY, maximum sustainable yield, and integrate another source of information and incorporate variables associated with the climate change. The research area is located in this 3 statistical area of the CCAMLR administration. Why? Because today this small place in the Southern Ocean concentrate the biggest krill fishery activity. Why consider this driver of the climate change? Because this driver has impact in the krill. This animal has intimate relation between its life cycle and seasonality timing in environmental conditions. This relationship is well described by Piñones and Federer here. In the early summer, when the krill spawn, their eggs migrate down in the water column, try to find higher temperatures to hatch. When the eggs hatch, they arrive under the sea ice to feed until the adult stage, principally in winter season. This performance, this timing between life cycle and environmental condition has been described in fishery science as catching match mismatch hypothesis, in which it indicates that any alteration on this timing with the environmental condition has effect on productivity in marine ecosystem, like ecosystem in krill. But the global warming scenario in Antarctic Peninsula is changing this timing. So we have a null hypothesis. In a climate change context, environmental conditions will have a negative impact on krill population in Antarctic Peninsula and will affect the long-term fishery management. What fishery data say about the krill population? Well, in this plot, we can see the long history of the krill fishery in 5 decades, where a change in distribution fishery activity is clear. In the recent years and the last years, this fishery has been concentrated within the Bracefield Strait, in a very intensive way, which has implication not only for the productivity in this stock, krill stock, but also do affect in the food web, because the krill is food for another species such as penguin and whale. In another hand, we also identify an explosive increase of the catch in this statistical area, in example 48.1 and 48.2. Of course, also affect the krill productivity, the structure and functionality of the ecosystem associated with the krill. The lens structure is another very important sort of information for modeling, and because define the process, both the recruitment and selectivity to the fishery. In this lens series and 3 statistical areas, we can see some years a very strong pulse of recruitment of juvenile are incorporated into the adult stock. This is important for the productivity too. Therefore, an integrated stock assessment model in krill aims to analyze and integrate different sort of information available in this krill fishery. An example, data from fishery that I just showed, from data from acoustic survey. This is data independent of the fishery. We have biomass or density. We need 2 life story parameters. It's very well described in different report or paper, in example, Tanasecos 2014. And the last one part is the climate change variable. An example, ice coverage, because have effect in the density krill, how we can see in Flores 2012. This kind of data represent a biggest challenge to integrate in stock assessment model. Briefly, this equation represents the state equation of the population dynamics. Looks like scary, but it's very simple to understand. In one hand, we have the abundance that depend of the recruitment and fishing mortality and natural mortality of the krill through the years and through the age of this species. Then we calculate biomass with the length weight relation parameters. And the next step, we calculate spawning biomass. The spawning biomass is the most important variable in this kind of exercise with the maturity OG. The stool-recruit relation is another submodel in this integrate model. In this case, Beverton-Hall. We apply the stock assessment model in 48 statistical sub-area. It's a preliminary exercise with the following pieces of information available so far. We have a time series of the catches from the fishery. We have 2 indexes. One index is CVE from the monitor fishery, and another index is biomass acoustics from the survey. And we have 2 very consistent data about the length of position from the monitor system, monitoring fishery survey. One way to diagnose the performance in a model is by fitting. Well, not just in this model, in all models. In this plot, we can see the length data from the length data from the krill in different years, and the green line represents the length model. Maybe in some years we don't have a perfect match between the predict and model, but we can check a good performance in our model. Finally, I want to show I think the most important population variable output in this stock assessment. The panel A show the fishing mortality, right, as increased in the recent years in a cyclical but continuous way. In the panel B, we have the recruitment, show the population pools through the entire time series analysis, which is a typical behavior of this kind of organisms, the short life and highly productivity, like a krill. And finally, this is a reproductive potential or spawning potential ratio of the population, in this case, the krill, which has decreased in the last year, probably by the intensity of the fishery in these years. And he's below, this level is below the reference level proposed in this work. To finish, some concluding remark. This research showed that it's possible to model the population dynamic on Antarctic krill through an integrated stochastic assessment model. It's possible to identify population variables such as spawning biomass, fishing mortality, and recruitment. We propose a biological reference point based on 75% at spawning biomass as proxy of MSY, and advise with this to the management. This exercise has preliminary result in 48.1 statistical subarea and show a population decline, which should be noted in a climate change context. This work is still in progress as part of Antarctic and Subantarctic Science PhD program in Universidad de Magallanes, Chile. Thank you so much for paying attention. Muchas gracias.
Thank you so much, Mauro. We do have some questions for you, but in the interest of time, we will save them for the discussion section. So thank you so much for that. Okay. Our next talk is a recorded presentation by Francisco Santa Cruz. And just to the IT folks, let's make sure we're playing the video entitled Spatial and Temporal Catch Concentrations for Antarctic Krill, Implications for Fishing Performance and Precautionary Management in the Southern Ocean. This is the pre-recorded talk.
Hello, everyone. My name is Francisco Santa Cruz. I'm going to present you the work Spatial and Temporal Catch Concentrations for Antarctic krill, implications for fishing performance and precautionary management in the Southern Ocean. This is a work from between colleagues from the Chilean Antarctic Institute. As you know, the Antarctic krill, Euphausia superba, is one of the most abundant species in the ocean, in the Southern Ocean. Antarctic krill plays a key role in the Antarctic food web. For aside, is an important food supply for marine mammals, birds, and fishes, and also contribute to move large amount of atmospheric carbon into the deep ocean. The Antarctic krill is an important fishing resource exploited by the largest fishery in the Southern Ocean. Most of the catches since the 19th occurs in the South Atlantic area of the Southern Ocean, in the Area 48, mostly in the Western Antarctic Peninsula, the South Horning Island, and the South Urea Island. All the fishing activities for Antarctic krill are managed by CCAMLR using both precautionary and ecosystem-based approaches in order to minimize the risk for the ecosystem. There is a fixed seasonal catch limit, known the trigger level, which is proportionally allocated among the sub-areas in order to avoid catches concentration. There is a common assumption that these precautionary measures and the high estimate of krill biomass implies that there is a low impact of the fishery on the ecosystem. However, the recent fast environmental change that we are noticed in the Antarctic Peninsula, which includes the ocean warming and an important decrease in the sea ice coverage, are factors that can impact krill productivity and this is spatial distribution. and thus are imposing new challenges for the krill fishery management for CCAMLR. Also, there is some concerns about how precautionary is the current management for these species under this environmental modification and the projected change in the Antarctic Peninsula. In addition to this, we also know that the krill fishery is an extremely aggregative activity which uses every season the same well-defined fishing hotspot. So we are working on evaluating the hypothesis that the increase of the seasonal catches and the persistent behavior of the persistent spatial and temporal concentration could determine locality depletion and could even impact on the fishing performance itself. To test this hypothesis, we use 38 years of catch and effort data to evaluate the fishing performance over time, and we investigate potential impacts of the spatial and temporal concentration on the krill CPUE. We use the CPUE expressed like a ton per hour, like an indicator of the fishing performance, and we calculate CPUE at the whole level, and then we estimate a mean CPUE per day per ship, and we calculate among the different small-scale management units in the Antarctic Peninsula. We know that the CPUE is a sensible indicator which varied depending on historical change in the fleet composition or the spatial distribution of the catches or seasonal variability within the season. So in order to reduce this variability, we estimate or identify the general trend of the CPUE. We use GAM models to standardize CPUE. And in order to identify factors that could impact the CPUE trend in time, we calculate two indicators of fishing concentration, one related to catch per area and other catch per day. And also we estimate the sea ice coverage in each SSMU. Among the main results, we identified that the sea ice cover has tended to remain stable among the studied sector, except for Elephant Island and Drake Passage, which has a decreased trend in time. We also identified that the fishing concentration has developed an increasing pattern, both in terms of spatial concentration and in temporal concentration. In general, the standardized CPUE showed a decreasing trend in all the evaluated sectors. We also identify a positive relationship between the standardized CPUE and the sea ice cover. In general, the standard CPUE was higher as increased sea ice cover, and we notice an inverse relationship between the standard CPUE and the spatial and temporal fishing concentration. In other words, as fishing concentration increases, the standard krill CPUE has decreased in time. Our results confirm the highly concentrated behavior of the krill fishery. The general pattern is that trawlers persistently operate in the small and well-delimited fishing hotspot, which recently have sustained the highest catches and during shorter fishing season, which is resulting in the highest spatial and temporal fishing concentration in 4 decades. The fishing performance has responded negatively to the increases in spatial and temporal concentration. To us, this is a concern, especially for areas in areas with decreasing trends of the CBE, which are very important for predators and for foraging during its foraging activities. We also think that it's very important to extend the spatial coverage of acoustic surveys and its periodicity to those critical areas, especially, for example, the Gerlach Strait, either by independent regular transects or using the fishing vessel as platforms. In particular, the Gerlach Strait had the higher levels of spatial and temporal fishing concentration during the last decade, and is one of the less monitored areas. So we think we potentially are fishing blind with unknown impacts on the ecosystem. And it's a situation which may be highly precautionary. Of course, we also notice that there is different source of uncertainty around the CPUE estimates and trends. There is some factor that could impact the CPUE estimation, like the krill flux or the krill reduction by predation by cetaceans. That are 2 factors that can reduce the local krill availability. However, we believe that CPUE is a valuable piece of information that should be used in complement with other information from surveys to inform responses of local krill abundance and its availability among the different fishing areas. I would like to thank the Marine Protected Area Program of the Chilean Antarctic Institute, and we appreciate any comments, observations, or doubts. Please contact us. So thank you very much for your attention.
Thanks so much to the IT team for a seamless video transition. I would like to invite Lucas Kruger for his first of 2 talks he's going to give during the session, but for your first one, Lucas.
Thank you, Cassandra. Let me share my screen. Okay, can you see it?
Yes, I can see it perfectly.
Okay. So I'll be presenting to you the results of the study called Projected Availability of Krill to Pygocellid Penguins in the Antarctic Peninsula. As you are aware, the Antarctic Peninsula is one of the regions of Antarctica experiencing the fast warming. And this warming takes as consequence the reduction of the sea ice, the increasing in the temperatures of the water, and reduction in productivity. And all of these joint factors have been taken to a decrease in the krill biomass and a change of the center of the distribution of the krill towards south. We have several lines of evidence showing that this decreasing of biomass in density is very likely. At least here I'm presenting results from the study of Atkinson et al. 2019, and the recent report on krill acoustics presented at CCAMLR that support the general idea of decrease in the krill biomass in the Antarctic Peninsula. not surprisingly, some of the most dependent predators of the krill, that are the pygoscelis penguins, have been on a decreasing trend in recent decades. Of course, there are some views that it's also a response of the recovery of the whale populations in the Antarctic Peninsula, reducing the availability of the krill for penguins, but it's believed that the climate change plays a very important role in those changes. So for testing how the density of krill would change according to future scenarios of climate change, we run a suitability model using a generalized boosted approach, where we evaluated if the krill density from the krill base is dependent on a series of environmental and climatic variables: the sea surface temperature gradient, sea ice thickness, sea current velocity, sea bottom temperature, and also the topography of the ocean floor, seabed depth and seabed slope. And we applied the generalized boosted model and found that the seabed depth and the sea ice thickness are the most important variables explaining the krill density on the Antarctic Peninsula. And when we projected it into the spatial information, spatial data, we could see that there's a tendency of the zones of density of krill move south, both in the west and the east of the Antarctic Peninsula. Also with some slight decrease in the South Shetlands and Elephant Islands, and an area of more or less stability in the Bransfield Strait. And also the modeled krill density was able to explain both the probability of occurrence of a penguin colony, but also the number of penguin colonies in the area. Here we have all the penguin colonies known for the area using the data from the MAPPPD data bank, and calculating the density on a 40 kilometers distance from the coastline. So here we see that both the probability of presence of a penguin colony and the density in blue are significantly explained by the model density of krill from our model. We also were able to identify a threshold where we have a higher probability of having a penguin colony, and also a value where there's a higher probability of having more colonies on a given area due to the higher density of krill. When this threshold is applied over the future density of krill, we can see here in blue the current krill density and the density in 2040 and 2090. So we can see that the south, both west and east, had an increase in krill density in future scenarios, but the other sectors of the Antarctic Peninsula had a decrease in the future density of krill. However, in the Bransfield Strait, there was a decrease, but this decrease never crossed the threshold that we identified for the occurrence and density of penguin populations. In the Galash Strait, this threshold was crossed only in 2020. However, in the South Shetlands and Elephant Island, this threshold was crossed even in 2040. So the presence and number of colonies of penguins match the predicted habitat of krill, and these expected changes in krill availability were consistent with current penguin trends. It is in the northern range of the pygoscelis penguin distribution in the Antarctic Peninsula. We are expecting we are already seeing substantial reductions in the penguin populations, and we are expecting to see even more steeper changes in the future. The question that remains is, can penguins persist under decreasing availability of krill? Of course, It will depend on the flexibility of the penguins to colonize new areas and their adaptability to shift from krill to other species. We know that from the 3 Pygoscelis species, at least the gentle penguin is more flexible, but the chinstrap and the Adelie in the peninsula are highly dependent on krill. More than 95 to 99% of their diet are the Antarctic krill. Therefore, these results indicate that they are likely to continuing being affected by this reduction in krill biomass. Thank you very much. That's it.
Thanks so much, Lucas. We'll see you back for your second talk in a little bit. But in between, I'd like to invite Catherine Gallagher to come give your talk. And just to say too, again, we're just going to hold all questions for the end. We do have questions for everybody, but we'll hold them for the discussion session. So Catherine, over to you.
Awesome, thank you. You can all see my screen?
Yep, it looks great.
Awesome, thank you. Turn that off. Hi everyone, my name is Catherine Gallagher. I am a postdoc at Stony Brook University working with Dr. Heather Lynch and also Mike Dinneman from Old Dominion University on a topic that's actually eerily similar to Lucas's, so I'll get right into it. I'm not going to reiterate to this group that Antarctic krill are important for Southern Ocean ecology, serving as a keystone species. And we all know that their distributions are patchy and populations are extremely variable and shifting, as we just saw along the West Antarctic Peninsula. Krill are macrozooplankton. They're slightly larger than your traditional zooplankton. And as a result, their distributions can be driven both by the ocean currents and the environment that they live in, as well as their behavior. We know they're capable of forming large swarms, as shown in the picture down here on the bottom right. We also know they perform diel vertical migrations within the water column. This allows them to interact with different flow regimes. Currents at the surface or close to the surface of the ocean are often very different from what we see at depth. And an example of this has been described in Palmer Deep Canyon. As part of my dissertation, I used models to describe a highly retentive subsurface eddy adjacent to the penguin colonies along the coast of Inverse Island. You can see the eddy shown here in this animation on the top. Through particle experiments and by adding diel vertical migration behaviors to the krill, to the particles we're adding to the simulation, we were able to illustrate that the addition of diel vertical migration behavior increases the residence times of simulated krill in this region to as high as 30 days when the migrations are very deep. And looking at penguin foraging regions in the area, we illustrated that this feature and the presence of this retention may help increase simulated krill abundance and availability to the penguins foraging in this region. So the overarching research question of my postdoc is, are penguin colonies along the West Antarctic Peninsula established adjacent to areas of high retention like we saw at Palmer and high accumulation of simulated krill? What we're hypothesizing here is that penguin colonies and the distance to different areas or grid cells where we're calculating retention and accumulation, they'll be closer to areas where these metrics are higher, resulting in this negative relationship. Really briefly, there's a lot of different methods that go into this. We're utilizing the Regional Ocean Modeling System, or ROMS, to release simulated krill throughout this domain weekly, performing a variety of different diel vertical migration behaviors. And we're simulating over one austral summer currently from 2008 to 2009. We're calculating those two metrics. We're calculating residence time using the e-folding time for each 40-kilometer grid cell shown here. And we're also calculating aggregation as simply the number of simulated krill within a 10-kilometer grid. And we're not counting those krill until they've been circulating the model for about 5 days. And we're going to be averaging these metrics over the Chicory period from December to March. We're going to be focusing using the mapping applications for penguin populations and projected dynamics, or mapped. We've identified penguin colonies in 4 distinct geographical regions, which are the different colored boxes here on the plot. We've identified at 210 Adelie Gen 2 and chinstrap colonies that have been surveyed at least once since 2006. So to get right into it, our results here, these are the results of residence time or retention in days across the entire peninsula. The different panels here illustrate our different migration depths, starting moving from shallow down to 25 meters in panel A, all the way down to 150 meters in panel E. And what we see here is there's not a lot of variation in between the different behaviors. We see patterns that stick out across the different behaviors. We see higher residence times generally in the southern and central regions of the WAP, as well as in the wet LZ, and not a lot of retention up in the South Shetland Islands and at the northern tip of the peninsula, which makes sense given its proximity to the Antarctic circumpolar current. Now, are penguin colonies located closer to these higher retention areas? So in this table, we've broken it down by our 3 different species of penguins, our different regions, and our different migration depths as the columns. NS in a grid cell simply means that the relationship was not significant in a linear regression. And the values illustrated here are the R² values of any significant relationships. And green colored cells support our hypothesis, meaning that the slope was negative, indicating that the penguin colonies were closer to high retention. And what we see here is that we see a lot more across all 3 species in the central and southern regions. Penguin colonies were generally closer to areas of high retention, and both Gen 2 and chinstrap penguins were closer to areas of high retention in the island regions as well. Now moving into aggregation, remember this is the number of simulated krill within a grid cell, this time on a 10-kilometer grid, so the grid cells are quite small. And here we do see some patterns as krill start to migrate deeper. You see more krill aggregated, especially along the central peninsula, when krill are migrating deep and retention is higher in comparison to when migrations are very shallow. But across all of these simulations, what really pops out is that there's a lot of krill in the island regions and along the tip of the peninsula. This pattern stays consistent. When we perform the same regression analysis again, we see that both Adelie and chinstrap penguins are closer to areas of high aggregation in the islands and northern regions. And then both Adelie, Gen 2 and chinstraps, all 3 species, are farther away from areas of high aggregation in those central and southern regions. So our conclusion so far, this project just got started in February. So what we've got so far is that Adelie colonies seem to be established closer to areas of high retention in the central and southern part of the peninsulas and high aggregations in the islands and in the northern WAP. Gen 2 colonies are established only near areas of higher retention. There was no relationship to areas, they were not close to areas of high aggregation in any region. And then chinstrap colonies are established closer to areas of high retention in all but this northern area of the WAP and high aggregations in the islands and north of the WAP. So we're still really combing through all of this data, but what is really interesting, at least to us so far, is that patterns are starting to sort of match where population dynamics of penguins. Adelies are doing well in the northern side of the peninsula, but not well in the central south regions where they're closer to areas of high retention, while Gen 2s are actively expanding south where they're close to areas of high retention. So that there might be something very interesting there. But if you noticed in all those tables, very little of the variance was explained in all of these relationships. Only about 7% of the relationship of the variance was explained. And we know there are other variables that play a role, such as available space, which is one of the things that we're going to be looking into. So next steps for this project, we're going to actually be using satellite-based estimates of diet composition to see if penguins are utilizing these areas of high retention or aggregation by looking at guano colors and satellite images. We're also going to explore different nonlinear relationships between these metrics using colony size or breeding success data from mapped and seeing if those are correlated with any of these metrics. And then finally building this, looking at this relationship across model years to see if it's persistent, not just in the one summer we've simulated so far. And that's it. Thank you.
Catherine, thank you so much. And like you said, it really builds on what we just heard about. And we will have some questions and time for discussion after Lucas's next talk. So Lucas, I invite you back to the stage for our final talk of the session.
Okay, thank you, Cassandra. I'm gonna share my screen again. Oops. Okay, I think you're seeing the presentation, right?
Yeah.
Okay, so let's go. Well, I'm going to introduce my second presentation, titled "Warming Precludes Lower Biomass of Antarctic Krill and South Shetland Islands: The Train of Consequences for chinstrap penguin Foraging and Breeding Success." These studies are from compiling results from the master thesis of Nuria Salmeron, as she is the first author of this study. Well, I took the opportunity to reuse figures from the previous presentation, and I won't go very deep into this. But as I said before, there is a high probability that the biomass and density of krill in the Antarctic Peninsula is decreasing in the northern part of the Antarctic Peninsula, and as consequence, the penguin populations are also decreasing on the area. And one of the mechanisms that it is explaining the reduction of the krill biomass and krill density is the reduction, the warming causing the reduction of the winter sea ice, and also affecting the productivity in the summer, as the ice melting in spring release a series of nutrients that feed the algae, so there's a higher amount of food for the krill itself. And if there is less sea ice during winter, therefore the productivity should be smaller in the following season, and we expect to have less krill, and therefore it will affect penguin foraging and penguin behavior and breeding success. So in order to test that, we tracked it in 2 different seasons with GPS loggers and time depth recorders, 25 individuals of chinstrap penguin breeding in the Harmony Point, Nelson Island, on the South Shetland Islands. We tracked animals in 2 different seasons, 2019-20 and 2021-22. And we had GPS fixes each 5 minutes and time depth records each second. We also calculated breeding success of 12 sub-colonies on the area by drone flights in the beginning of the season in early to mid-December, and in late January or early February to estimate chicks and calculate a measure of chicks raised per nest. Okay, here we have a kernel distribution of the penguins in the first season in blue, and the lines represent each foraging penguin in the second season. And by a first inspection, we can see that some penguins foraged farther from the area that the penguins foraged in the first season, giving an indication of the results. So from several parameters, trip parameters that we calculated, we found different differences between seasons for foraging trip duration and foraging trip cumulative distance, meaning that in 2021 the birds forage longer and farther, not farther, but for larger distances. The frequency of complete dives were slightly smaller in 2021. The cumulative dive duration, it means the whole time they spent in diving in the bottom phase of the dive, was higher in 2021, sorry. And also the maximum depth of the dives were deeper in 2021. Finally, the mean capture effort during the dives were also higher in 2021-22. The capture effort was calculated at the number of wiggles that the animals would perform during the bottom phase of the depth. And the breeding success was 1.4 times lower in 2021 compared to 2019. And well, we also quantified the variability of environmental variables within a 75-kilometer radius from the breeding colony. And when we compare the winter sea ice cover and the summer productivity, we found that both were lower in the second season compared to the previous one. And we also conducted within the area where the penguins foraged, we conducted acoustic samplings for krill biomass. And we also found that in the second season in blue, not only there was more krill, but also the krill was closer to the surface. As you can see in the red line, this peak of biomass of krill was around 75 meters, and in the previous season it was below the 5 meters depth. And of course, when we check that or compare that with the behavior of the penguins, the mean dive depth per trip and the total capture effort per trip, we also see a change that is coincident with these differences in krill density and krill depth. So the results that we found support the idea or the view that increased foraging effort is a common indicator of lower resource availability for central place foragers. And furthermore, the low breeding success is a result of a low resource and probably increased foraging costs. So we have to ask ourselves in this recent context of rapid decline in Antarctic sea ice caused by the rapid warming of the Antarctic Peninsula, if we are going to have more frequent episodes of low ice, is it going to be translated in low krill availability for the penguins? In this context of a krill, reduced krill availability for penguins and increased consumption by whales and increased consumption by the krill fishery, we have to take into consideration all this scenario and all these probable effects that the climate change and environmental changes in the Antarctic Peninsula might cause to the penguin populations. Thank you very much.
Thank you so much, Lucas, and thank you to everyone for their great presentations today. I'm going to pass it over to Jilda for the start of our discussion.
Thanks, Cassandra. And so just to give everyone a heads up, so for this discussion portion of the session, the idea is that we're going to give everyone in the attendees section the option to become a panelist. I encourage all of you to do so, even if you don't want to say anything or turn on your video. You might change your mind and it'll make it a lot easier for you to interact with all of us if you do so. So Cassandra is going through everyone now, giving you the option to promote to panelists. You just click yes, and you'll show up in the panelist section, which means you'll be able to use the chat, you'll be able to raise hands, turn on your video, talk, all of that. So feel free to take up that option. I want to kick off the discussion section with a point that's almost a bit unrelated to any of the talks actually we had today, and it was something I just wanted to point out. I don't necessarily think we need to come up with a solution right now, but I just wanted everybody to roll this over in their heads kind of moving forward. And it's a equity, diversity, and inclusion point, EDI or DEI as it's often called in the US. For this, over the two sessions, we had a total of 14 talks, and only 4 of those talks were from women. And, you know, it's not like we rejected all the women, and it's also that we don't want to have talks from all of the wonderful men who have been speaking here today. But I think that this is emblematic of a bit of an issue that we have in a lot of the fishery sciences about having this imbalance between the amount of female and male scientists that we have engaged. I mean, of course, we have 2 females convening here, which is very exciting, but I would have been excited to see more females bringing in abstracts to give talks. And so I think that, I don't necessarily know what the solution to this is. I think we all just have to continue to promoting our female colleagues that are doing this kind of work and elevating their voices. And I just wanted to throw this out there for everyone to kind of go home from this session to sort of be thinking about this, and just so that we're all aware in this sort of fisheries management arena that this is a little bit of an issue, and we have to try to be creative to think about how we can make this better, essentially moving forward. So obviously, if anyone has any thoughts on that, feel free to chime in, but I'm not necessarily meaning to start a discussion on that, but I just wanted to throw it out there. And so in the meantime, I will pass actually the baton over to Cassandra to bring up another point that we wanted to discuss with all of you.
Thanks so much, Jilda, and thank you for highlighting that. Yeah, so I have promoted everyone who's in the session to panelists. A lot of you declined. If you didn't mean to decline and you would like to be promoted, please just message us and let us know. But again, we're not going to force you to sort of come to the stage and have this discussion with us. I think one of the things that is really striking me about listening to all your talks is thinking a lot about, and especially in lines of our session, we're talking about management implications, and everybody's work that was presented today certainly has really strong management implications. And I would personally love to see all this work come forward in the CCAMLR sphere. And so that I just want to throw that out, that would love to hear ideas on that. And one thought that we've been thinking about in the context of SCAR, in the context of this meeting, but also SCAR's role. So you may know that SCAR has a delegation at the CCAMLR meetings, and I have the great privilege of sitting on that delegation. And part of our role is to bring forward science to hopefully inform management. And a lot of times we do that through the existing SCAR groups, and we're always looking for ways to do that better. Like, how do we, and we try to stay on top of the research, but obviously there's so much research coming out constantly. But the research here certainly has strong management implications. So I'm really keen to hear from this community, panelists and attendees alike, any ideas you have about how we can better bring forward science to CCAMLR through SCAR or through other means, and to just hear everybody's ideas about that, how do we increase that flow of science into management? So great. So maybe we can try to promote one more time, folks. But yeah, I invite anyone who has a thought on that, panelists or attendees. I think some of you probably think about this.
Feel free to raise your hand if you don't want to just jump in. We'll be happy to call on you. I might start the discussion with a brief idea that actually came up when Christopher mentioned this at the AM session, which was, as some of you may have known about or been at, there was a mini symposium earlier this week with regards to the science policy space, but focus a bit more on the Antarctic treaty system. And the purpose of this workshop was sort of bring scientists who are less familiar with this space to kind of make them more aware of how they can bring their science into this space, how science is taken up by these type of policymaking bodies. It was, in theory, geared towards early career scientists because these are the largest body of scientists that might need this type of information, but obviously there's many mid-career and senior scientists that don't necessarily have that are not necessarily familiar with this space. So obviously it was, I think, very useful to everyone who was involved. I unfortunately wasn't able to attend, but I was involved in parts of the planning, so I did see that this was being put together. In any event, my point is that having a type of workshop in this vein at a future SCAR OSC that was focused on CCAMLR in contrast to the Antarctic Treaty System and focused on Southern Ocean work, that could be really useful. And this could be an initiative that's sponsored by SCAR and CCAMLR, for example. It could be in the context of an OSC. It could be a totally separate workshop that happens outside of the context of the OSC. So, I mean, this was just brainstorming when Christopher brought this up in the AM session, but in case anyone has any thoughts about this.
Graphic novels go to all the delegations, Marina, please.
Hello to everyone. Nice to see you here. So one thought that comes to my mind is I'm from Argentina, a developing country, and so we always speak of fostering the integration of science into policymaking. And there is one thing for developing countries, that is democratization of science, and we don't have the same access to, or the same possibilities to access science as developed countries. And that's a thing that probably makes us more difficult the task to integrate science into policymaking, not because scientists do not know how to speak in English or how to get to published papers in scientific magazines, but the technicians at policy bodies do not have access to that, or do not know how to get to that. So the democratization of science is one issue that maybe through SCAR might be started to be discussed, or try to see how to foster getting publications into other languages, or free access to publications, or many things that might be worked as, or solutions to be thought to be more inclusive in regards to that. Thank you.
Thanks, Marina. I definitely agree with all of those ideas. Let's keep in mind how to do them. David, go ahead. You can unmute and let us know your thoughts, and welcome.
Well, I'll get on my soapbox. CCAMLR was created because there was fear that a krill fishery would impact the recovery of whales. And it just keeps blowing my mind that there is no systematic survey to determine the abundance of whales along the Antarctic Peninsula. The last survey was done in 2003 by the Japanese circumpolar effort. Whales, I think people are afraid to know what whales are, what their impact is. But to me, they must be the largest fishery out for krill along the Western Antarctic Peninsula, and they're increasing. They were increasing, particularly humpback whales, by an astounding rate up through the early 2000s when the last series of surveys was done. So it just blows my mind that CCAMLR hasn't gotten on a soapbox as well. Again, I think people are afraid to learn what the impact of whales potentially is or is on krill abundance. Okay, off my soapbox.
Thanks, David, for your thoughts. Keen to hear from other folks, and again, certainly from attendees as well as panelists, and all of you panelists certainly spoke very elegantly with highly relevant work. And if you have even your own thoughts of how do you help facilitate your work into the policy space.
May I talk?
Please.
Yeah, okay. So, well, I had to add some points. I support what Marina said about how kind of it's quite more difficult from undeveloped countries to reach SCAR groups and to get involved, and also maybe the way how our scientific councils evaluate CCAMLR reports, or how CCAMLR reports are rated within the scientific community. That might be a point to tackle maybe in a future. So, and on the other side, I wanted to share some thought with David. It's a pleasure to virtually meet you. There's an I don't know if you know Andrew Lofter from the North Polar Institute. He's planning a CCAMLR project, I believe so, that is going to happen this next season, in which they will use the tourist vessel platforms to start this whales census again. And well, I believe it will be working for 2 years, but that's just a comment. That will also provide new insights on whales distribution and so on. Maybe I wanted to change the tone and go back to some lectures. I wanted to ask Thomas, we already discussed some things of his work, but I don't know if you consider how could the tourism be involved in the possible outbreak. You know, Endeavour Bay, Paradise Bay are highly tourist hotspots. If you consider that as a possible input of these kind of parasites, or I don't know, what are your thoughts on this regard?
Yeah, that's definitely something we've been thinking of. Could this parasite been brought in by tourist vessels or other type of vessels, actually? Because all vessels discharge some salt in the environment. So the problem is that we don't know where this pathogen comes from. It could have been there actually already for a long time. The fact that it's a new genus that has not yet been described anywhere or found anywhere else on the planet led us to think that maybe it's just something that is already in the environment and maybe not linked to tourist activity or human presence in general. But we can't exclude it. And the more we're going to know about the different pathogens and parasites that exist, the more we will be able then to try to connect dots and see whether maybe in South America, maybe we can find the same parasite. It was originally found in aquaculture settings and fisheries in Norway and Iceland and this area. So maybe with all the industry in Chile, for example, with salmon, if there are some outbreaks, maybe we will be able to identify the parasite and see whether there could be a connection between oceans, between different parts of the world, and then what could be the drivers that bring those new pathogens across.
Monica, you wanted to ask a further question?
Yes, thank you. And I'm sorry for bringing up so many different issues at once, but it's different. Every time you get to a different meeting, you have a different protocol. But it was very nice to know what the rules were. Anyway, Thomas, I was wondering if you guys had done any genetics of the viruses, because we did see a few presentations over the past few days where they tried to track the origin of the viruses and were able to make connections to North America and Europe in different subjects with different species, but they were able to track by doing genomic analysis. So I was wondering if it's something that's worth doing to see the source, because what you reported is quite concerning, because there is a 30%, it's not trivial. And if you can try to assess where it's coming from, it might help a way to prevent it or simply to exterminate it. Thank you.
Yeah, absolutely. Thank you very much. We did some genetics. We don't have a genome, complete genome. We're working on it. It's in the works. But for now, we were just able to use the 18S DNA gene, which showed us that actually like the parasite we identify is really different from the ones that are known from flatfishes and salmons, and also very different from like the classic XL parasite. That actually one of the sequence we used was sequenced from a specimen caught in the Ross Sea, and so it's really a group that is well individualized. So it's really hard to tell where it comes from. We just know that it's related to those other groups, but there is so far no other closely related organism known or described around the planet. So we can't really track yet where it comes from. But if more people, if when people capture fish with disease, and that was kind of the same before, until we stumbled on so many of them, I was kind of like overlooking those specimens because I was like, well, this fish is diseased. What I'm going to get from it is probably not, is probably biased. So I was kind of like discarding those fish. But now I'm much more aware, and I hope more people will be aware as well, to actually make a point in like actually trying to sample, even if it's just a little bit of the sample in ethanol. We can do a lot of things with that. And so if people are aware of it and know what to do with it, then we can then creatively increase our knowledge and maybe then be able to track to see whether this parasite is native and endemic of Antarctica or not. One of the big problem too is that we ignore pretty much everything about the life cycle of those parasites. Nobody has ever found dividing cells. We don't know if the life cycle is yearly, if it's every 20 years, like I was saying. It's very patchy in any case throughout the world. So we don't know the reservoir, we don't know life cycle. There's so many things we don't know. So we need to go back and look at the sediments, look at more species, systematically search for wounds and disease signs to try to really enhance our knowledge, because it's really an overlooked aspect of Antarctic science so far.
Thanks, Thomas, for that feedback. I actually had an additional question for you, but before I do that, I wanted to pass to Lucas in case you wanted, because you were talking about maybe wanting to make an intervention. So Lucas, you have the floor if you'd like, otherwise.
No, I'd like to go back to krill and penguins. So you just end the discussion on this and I come back.
Lucas wants to go back to krill. Okay. It's understandable. So thank you, Lucas. We will do that shortly. Let me just pass on then this additional question while we're talking, Thomas, about your work with the parasites. So we had sort of two questions. One was the extent to which you think that this might be something that we might be seeing in other species of notothenioids. And I know that you looked at differences in growth rates between diseased and non-diseased fish. But I guess the ultimate question is, will there be survival or life consequences, well, or rather survival consequences for these fish? And I'm not exactly sure how you would test that per se, maybe looking at the differences in ages between fish that are diseased versus not, and maybe all the diseased fish are much younger. But yeah, just to hear your thoughts on, I guess, how diverse this type of parasite infestation might be among notothenioids, and also ways in which we could see the impact on longevity.
Yeah, so how dispersed, we don't really know. I did all the literature search on the subject, and other species of Trematomus have shown to be affected in the Ross Sea. So Clive Evans, for example, like published about those before. And honestly, whether there would be life consequences of the infection, I would say yes. The fish were really, some had like massive tumors. Even if the tumor resolves by itself, like which I doubt, I would imagine that the fish is not doing well. Maybe it's not going to be killing it, but I would imagine that then the investment in reproduction would be already like dramatically reduced, which then would have consequences on the recruitment and the maintenance of the population. So yeah, the main question is we need to know more. The thing, when we were doing that, when we captured those fish, we were not ready for capturing those. We were not looking for that. We were only 3 that night, fishing and sorting through hundreds of specimens. And we could only keep a certain amount of specimens and not everything. So our view of what was happening is also already fragmented. So that's why we need more data, go back and do a much more systematic survey, keeping every single individual, try to explore adjacent areas to really be able to understand better. And for about aging is really something we would like to do in the future, because maybe there's also an age threshold where like fish don't get affected before they reach a certain age. So, and of course we want to also to see how the infection impacts reproduction, which we have not been able to do.
That makes sense. Thank you so much, Thomas, for that feedback, and again for your talk. Because we're 3 minutes to the hour, I'm just going to do a quick housekeeping announcement before I pass the baton to Lucas and we go more into the krill discussion. So IT team, correct me if I'm wrong, but I think we can go a little bit over. I personally can stay until 15 minutes past the hour, so I think we can envision potentially extending our discussion to that point. I understand some attendees and some speakers may need to leave earlier. And just a reminder to everyone that you can contact any of the speakers via email for further questions. This whole session is being recorded and will be available on YouTube. We as the conveners will be following up with all of the speakers with information about where you can store your talk, where the recording links are, and so on. So just remember, anyone that has to leave a bit early or what have you, there's many ways to continue to ask questions to the speakers, continue these discussions, and so on. So let's hope to go to about 15 minutes past the hour, unless IT tells us otherwise. And Lucas, I hand you the floor.
Thank you very much. Actually, I'd like to comment on some of the results of Virginia about the silverfish. And I was thinking that it might be very interesting to use it as a parallel to what have happened with the krill in the Antarctic Peninsula, because we have a free-ranging predator being reduced by the fishery, and then you have more silverfish for the central place foragers. Maybe this, as I said before, the silverfish surplus, like the krill surplus, maybe help us understand what how this mechanism works in the. And of course, as we know, the whales are now on the rise. Maybe the similar would happen if we stop the fishery of the toothfish. So just want to comment on that maybe it's possible to have a parallel mechanism that can explain the other mechanism.
Yeah, I think it's I really like that there are similar mechanisms. I really think that they can be comparable. The thing is that we were using the big toothfish catch by the fisheries as an index of the quantity of the silverfish. So we don't really have direct data of as far as I know, of the quantity of silverfish that we have. So that would be like an important step to try to measure the silverfish on the Ross Sea and to see if the index that we consider is right and our hypothesis is really right. So because of that, I think we need more research knowing exactly the bottom of this trophic chain, what's happening there.
Thank you.
Thanks so much,
nice work.
Lucas, anything further to comment or say?
Oh, no, thank you. I'm okay.
Cool. Thank you so much. And thank you, Virginia. Monica, I see your hand. Go ahead.
Thank you, Cassandra. Well, it was really nice to see these talks, and I would like to make a plea to biological and physiological studies to complement what is being done, because you can clearly see isotope signatures reflected on the predators if you do read, if the changes that we are portraying here really carry out through the food chain. So that is one important aspect. And the other one is we are getting to a point where a lot of automated studies are being pushed and are flourishing, but we cannot forget the biology because everything we do is based on previous knowledge. We need to keep the biology up with these changes to make sure that they reflect what's going on with the individuals. So tagging, tag reciting, sampling of a few key colonies is very important so that we can support the theories that are being put forward. Thank you.
Thanks, Monica. I totally support your plea that hopefully we now have all heard. Thank you for that. David, go ahead. I see your hand.
Well, first, I wanted to congratulate Tomas for including Arvind Virasani in his group. Very amazing virologist. But also, in regard to krill, I've done a lot of work in the California current as one of the eastern boundary currents. And these eastern boundary currents, knowing Peru, South Africa, and North Africa, they all are dominated by anchovy and sardine forage fish. And I've always been looking at to see if there's some sort of Antarctic analog. And so I was impressed by the first couple slides that Manuel showed, in which he summarized the natural history of, or demography of notothenioid fish. And so it impressed me that, well, in the California current, okay, there's lots of sardines and anchovies, and they're important forage, but also there's this benthic family of fish called rockfish, genus Sebastes. And it seems to me that the California current is more the analog of, let's say, the Western Antarctic Peninsula than is, at least from what I know about the Peru current or Benguela current, which don't have a family of fish like Sebastes. And so like these notothenioid, these rockfish take a long time to mature, like into their teens, and they live a long time. And they, back in the 60s, they appeared to be incredibly abundant. And so there was the fishery agencies had no take limits. And so they depleted these fish quickly. And the same thing has occurred with the notothenioids along the Antarctic Peninsula. And I guess for the same reason. And so, anyway, I was just struck by that parallel between the California current and the WAP. But also in regard to these the anchovies and sardines, in the California current, which I think is the poster child for ecosystem-based fishery management, the fishery managers actually reduced the sardine fishery because they anticipated sardines becoming depleted because of changes in the climate. And yet I see CCAMLR not paying attention to climate change in the Antarctic Peninsula. Well, so that, anyway, I don't know where I'm going on this, but just these parallels between not just the biology of the California current versus the WAP, but also I would hope that some management lessons could be taken from the fishery management in the California current and applied to the WAP. Okay, I'm done.
Thanks so much, David. I mean, I feel like you brought up some good questions that I would love to throw out to the group. And it's certainly things that have been on my mind of how do we better manage for climate change. And this is certainly something that SCAR as a group also is trying to continue to push forward. What are the climate change implications in the Southern Ocean and how to consider them. So I'd love to hear other folks' thoughts on this. How do we better manage for climate change? And I guess that also there's this obvious question associated with that. A lot of you were talking about potential impacts on krill and their predators, some of these other potential impacts on fish and the ecosystem, and yet fishing continues. So maybe there's even a bigger question of can we continue to fish at the levels we are given climate change impacts? And so I'll just throw that out there. And Lucas, I see your hand.
Thank you, Cassandra. Well, I don't think it's fair to say that CCAMLR is not concerned. Of course, I agree. CCAMLR is answering very slowly to this, but CCAMLR is trying now to implement a change in the management, using a feedback management that should be more dynamic and therefore change the amounts of catch and where to catch in accordance with the variability of the area. But it's not like it doesn't care. CCAMLR doesn't care, but yes, it's too slow. It's definitely slow. The improvements should be already being implemented, and well, maybe we should try to speed up the pace and make some pressure for the decision makers to be more active and, well, make their part.
Thanks, Lucas. Manuel, did you want to respond?
Yeah, I'm sorry. I don't know how I lost it.
You're good. Go right ahead.
I'm sorry. Yeah, well, I agree with both sides of these thoughts. I believe that, and this comes back to what Monica said, at least in what reproduction in fish is concerned, we do not know how is temperature and photoperiod, as I mentioned in my slides, how are they framing and shaping when and where fish species are reproducing. At least what for example, CCAMLR has been focused on commercial fisheries or commercial target species. I mean, do we know why toothfish goes to the seamounts? Or what's going on there in terms of environmental changes they are facing to make these areas, or why are these last discovery of the ROV at the Weddell Sea are there? I don't know if you were aware when reading that paper, the inside this vast breeding colony, there was water temperature was 1 degree higher than the other ones. So it comes back to my mind, are there hot spots of water temperature in deep waters? How fishes are searching there, and at this paper also they could find a former nest. So there's a site selection, and what do we know from basic science? Where are the tipping points in terms of temperature or at least in fish reproduction, which is really important, and how it's CCAMLR going or managing fisheries considering this previous knowledge.
Thanks so much for that, Manuel. Jilda, please.
Thanks, Cassandra. I wanted to come back to David's comment, and I'm sort of responding in a bit to, I guess, the group as a whole, because I don't feign, David, to have a modicum of the experience that you have in this arena. I guess I'm thinking with the idea of comparing the management in the California current, and to be fair, this is also something I don't know. Is this a decision? Are these ecosystem management decisions made on the scale of California, or are they made on a federal scale, for example? Because I would almost compare the slowness of CCAMLR to the slowness of the US government, for example, and trying to make 50 states or 50 senators come to an agreement on something. Whereas if you have California, which knows what it wants to do and it can get it done, you know, and so I think you're obviously aware of this, and I think it's just an interesting thought process to think about how, you know, processes, why CCAMLR is so slow and what can we do to make it better, because we have scientists from the US delegation who have these experiences of how management has been done in certain places in the US, and also other delegations that could bring this to CCAMLR, and why isn't this being taken up? And I think obviously it comes back to political issues, which, you know, is the analogy between a federal US decision versus a US state decision. It would be the same, one country in CCAMLR versus all the countries in CCAMLR. And I think just, I think this comes back to the interest of having, I don't say this for you, David, because I know you know this, but I kind of an interest for the community of SCAR to be understanding better why is CCAMLR so slow, right? And I don't necessarily think we as the community ground up can unilaterally change that. There definitely needs to be top-down changes in the way CCAMLR works as well. But I think it would be interesting to work on both sides. And, you know, again, coming back to this idea that Cassandra brought up in the beginning of having some sort of workshop, or trying to have more of these types of workshops to bring in early career researchers, to bring in more of the pure biologists that don't necessarily know how this work goes into CCAMLR, or might not understand why very basic things like taking climate change into consideration in management are taking so long to take effect, or why it's been 10 years since the circumpolar MPA network was proposed and it's never been ratified. You know, these are things that if you're not as involved in the process, and I don't necessarily feign to be an expert, I'm myself learning as well, that this doesn't seem to make sense. And I think we don't necessarily have the answers here, but I think it's a really good discussion to continue to have in the community, and to bring this more into SCAR, to kind of, you know, why is this happening and what can we do to help, and just bring more people into the conversation. So that's just my comment on that.
Jilda, thank you so much for that. And certainly if anybody wants to respond, I welcome you to. I will just say, yeah, I think you're really right that the scale matters, right? And the politics matters, and that's why things can move slow. On a positive note, I would like to maybe recall that there are moments when CCAMLR has responded rapidly. When illegal fishing was rampant, it was pretty quickly that CCAMLR was able to respond. And so what I might push is that maybe we can all do a better job of showing how urgent climate change is, like how urgently we need to respond to it, that it sort of has that impact of, because that was it, illegal fishing had such a big economic impact on some countries, and maybe if we can do better of showing, yeah, climate change is going to be worse than that and need to respond to it. David, I see your hand, and then I think this will probably be our last comment, and then I'll pass it over to you, Jilda.
Okay, I think CCAMLR acted so quickly on illegal fishing because certain members were seeing fish taken that they wanted to take. So it was easy for them to help, you know, do something about it. But I think the reason, well, in terms of the California current, it's managed by the Pacific Fisheries Management Council. So it's not just California, it's the whole West Coast. And they manage things based on science, pure and simple. But also there's a very involved, you know, public along coastal West Coast. So I think those two things resulted in very effective fishery management. You know, the fisheries got burned by the demise of sardines back in the 1950s and 60s, and they learned from that. And they created this multi-agency group to, you know, conduct studies and, you know, on these fish dynamics. And now more recently, they're applying what they found. And I think I said, you know, the sardine fishers screamed bloody murder when when the fishery on our sardines were curtailed, when there was still a lot of sardines to be caught. But the fishery managers knew that because of the changing climate, ocean climate, that sardines would start to decrease. And, you know, so basically it was science that proved to be effective. Okay, so anyway, just a little background.
Thanks, David. Jilda, I'll kick it over to you for closing remarks. And before I do that, I just again want to thank everybody for being here, for their wonderful presentations, for the discussion. And again, all of this has been recorded, so we can certainly share it very widely. Jilda, over to you.
Thanks, Cassandra, and thanks, David, for that last comment. I also found it interesting to kind of understand a bit better how management is working in the California current. I think the point you mentioned about outreach, for example, made me immediately think of Thomas' graphic novel. I feel like if countries could inform their public better of the value of the science that they're doing, they would vote for people that make the policy that will base that on science. Of course, voting also has to work in their country, so it's a big ask, and I think we get into much bigger conversations. But in any event, I think it's all food for thought, you know, that we can mull over as we leave this session. I wanted to mention also some of Monica's comments in the chat with regards to how to uptake science into CCAMLR. And you know, she mentioned working papers, which is of course true, and I think many of us are aware of that. Just in case there's people that aren't necessarily on that page, I wanted to throw in the link to the list of SC representatives to CCAMLR. So if you're ever interested in getting your work to be discussed at a CCAMLR working group meeting, all you need to do is contact a scientific committee representative of your country, and they can present your work at the working group meeting. Or they can even let you come to the meeting, either hopefully in person coming soon, but it's been online the past couple of years. So just to be aware that that connection exists between what am I doing, what am I presenting here at SCAR, and what do I want CCAMLR to hear about. Yes, so I think we're a little bit past time now, and I want to let everybody go. I want to just reiterate Cassandra's thanks for everybody for their talks, both in this PM session as well as for the folks that were there, the AM session as well. You know, we're really, as we did 2 years ago, but I think we'll try even harder to not let the discussions end here. There will certainly be follow-up from us as conveners with information about sharing presentations, everybody having each other's contact info, about when the recordings will be available. And I also think discussions about such a CCAMLR policy workshop to the SCAR community as I think something that should move forward between now and the next OSC. So again, thank you to all the speakers. Thank you to Cassandra. Thank you to Christopher, who has helped me in the AM session to convene. Thank you attendees for participating and joining this discussion, asking great questions to our speakers. And I wish everybody a wonderful rest of your day and evening, and that I'm excited to keep talking about fishery science with you guys for years to come.
Thanks so much, Jilda. Thanks, everybody.
Thank you very much.
Thank you. Bye.
Bye.
Thank you so much.